Hello everyone, and welcome to D1.2, which is protein synthesis in IB biology. Let's get started. So first of all, this slide might be the most important one in the entire IB biology curriculum. So when I ask my students, how does DNA actually have any effect on the characteristics of an organism, most of them actually blank. So that's through gene expression, right? So genes, DNA, code for proteins, and the proteins then determine the characteristics of the individual. So for that, we need transcription and translation, right? Transcription is turning DNA into mRNA, and then translation is taking the information from mRNA and turning it into an amino acid sequence or a polypeptide, which then folds into a protein, right? And that happens in the cytoplasm. Importantly, transcription can be switched on or off, meaning different cells in our body produce different mRNA at different points, right? So for example, the gene to make insulin is only expressed in the pancreas. So different cells have different transcriptomes, right? The transcriptome is the full range of RNA made in a cell. If you understand this, a lot of biology will be way easier from now on. So now we're going to look at transcription and then translation. So first, transcription is the synthesis of RNA using DNA. We just talked about that. How is it done? It's done by RNA polymerase, okay, which is an enzyme. So RNA polymerase you remember how in DNA replication we had tons of enzymes. We had helicase, we had DNA polymerase, we had ligase, blah, blah, blah. RNA polymerase is a machine. It does so many things, right? So RNA polymerase first unwinds the double helix, right? It doesn't need helicase, it does it itself. It then positions RNA nucleotides to do complementary base pairing, and it then links them to form a continuous strand through covalent bonding here, right? And finally, at the end, it detaches the assembled RNA. Importantly, when we're doing transcription, we only used one strand, okay? We don't use two strands. That's actually quite an important thing to remember. So we have the sense strand, but we also have the antisense strand. So the one that the actual RNA nucleotides bind to is the antisense strand, and then therefore the mRNA has a very, very similar sequence to the sense strand, with the main difference being that thymine is substituted by uracil, because remember, RNA does not have thymine, okay? That's super important. But everything else is the same. It's still complementary base pairing, right? It's because of the hydrogen bonding. Good. Also very important to say, it probably goes without saying, but the DNA sequence does not change at all during transcription, right? So DNA templates can be transcribed many, many times during the life of a cell, and the DNA does not change. Okay, so now on to translation. But before we do so, I want to briefly mention that all of these slides, including for all topics and all themes, and very, very extensive notes for all of the slides, are now available on my website. If you're unsure, you can get a free sample, so go check that out. But now let's move on to translation. So, translation. As we just said, translation is taking mRNA and coding it into an amino acid sequence. Now, how does this happen? Okay, mRNA, remember, is going to contain a sequence of bases. It's what you see here, right? This green thing. So it's going to be A, U, C, G, a sequence of those. Now, how does that work? It works through codons. And you might be saying, what is a codon? So a codon is a sequence of 3 bases on the mRNA. Why do we use a sequence of 3 bases? Why? Because that's not too obvious, right? Why not use 2 bases? Okay. If we used a code with 2 bases, so we could use duplets, right? That would only give us 4 to the power of 2 combinations, right? Because we have 4 bases. So we would only have 16 combinations, which is insufficient because there's 20 different amino acids. So instead we use triplets. Codons are 3 base codes, right? Because 4 to the power of 3 is 64, right? So these are the 64 different combinations. We would only get 16 of these if we had 2 letter codes. So this way, we have a code for every single amino acid, but also super importantly, it allows for redundancy, okay? Redundancy is a very important term to remember. It means that there's multiple codons for every single amino acid, okay? As you can see here, most amino acids have multiple codons that code for them. So then how does it actually work? Okay, so it happens in the ribosome. The ribosome is found in the cytoplasm. It can be found by the rough endoplasmic reticulum or just free in the cytoplasm. And so the mRNA is going to bind to the small subunit of the ribosome. And then the large subunit has 3 binding sites for tRNA. And then it also has the enzymatic activity to form peptide bonds between different amino acids, right? What is a tRNA? Okay, so a tRNA has an anticodon at the bottom. So basically the anticodon is complementary to the codon, okay? Here's where things get a bit complicated. But if the mRNA is AAA, then we're going to have an anticodon, which is UUU. And then the tRNA, apart from having the anticodon, it also has an amino acid attached to it. And the amino acid corresponds to the anticodon, right? So this is how we actually decode the DNA, right, in the end through mRNA. It's codon-anticodon pairs, and then the anticodon carries an amino acid with. So this ensures that the correct amino acid is added to the growing polypeptide chain. So as I just said, the genetic code is redundant, but some other things it's degenerate, right? But some other things that you need to know is that there are start codons and there are stop codons, okay? So that signals where to initiate translation and where to end translation. And something else that's super important is that the genetic code is universal, so it's shared by all living organisms with very, very few exceptions. Some more things that you need to know about translation. How does that actually work? So it's in repeating cycles, right? So one amino acid is added at a single time, okay? How it works is the ribosome moves one codon forward along the mRNA every time. And basically, as you can see, there's always two ribosomes on the mRNA, on the ribosome, right? And then one new one comes in, displaces the final one. Then this one in the middle is carrying the entire polypeptide chain. This new one comes, then the phenylalanine in this case is bound to the asparagine. So these two amino acids are bound together. So now the polypeptide starts to be on this tRNA, and then the ribosome can move again. So another one will come, etc., etc., etc. Okay? So hopefully that makes sense. A tRNA comes, another exits, right? And in between the ones that are in the ribosome, you fuse the amino acids together to start making a larger polypeptide chain. Any questions about this, please do leave it in the comments. And last thing for those in SL, gene mutations. So a gene mutation is a change to the base sequence of a gene. So we're talking about DNA again, okay? Important. Specifically, a base substitution is a type of mutation which changes one base only, okay? And that changes the codon, and that can change the amino acid or not, right? It depends, because remember, the genetic code is degenerate, meaning a lot of different codons code for the same amino acid. But one example that you need to know is sickle cell anemia, where basically there's a change from CTC to CAC, and that causes a change in the amino acid, glutamic acid to valine. That means that hemoglobin now is faulty, right? So the red blood cells have this weird sickle cell shape, and that basically leads to the red blood cells clumping, right, in capillaries, which can lead to anemia, which is where you don't get enough oxygen. However, interestingly, okay, sickle cell anemia is much more common in parts of Africa. How come? Because sickle cell anemia actually protects you from malaria, right? And malaria is very, very deadly. So it is worth, in a lot of cases, having sickle cell anemia, right, just to protect you from malaria, because sickle cell anemia is, in most cases, not deadly, okay? It's an evolutionary advantage in some parts of Africa. So moving on to the HL part. First, you need to remember that transcription and translation are unidirectional because of enzyme specificity. So they're both 5' to 3', the same as DNA replication. I think that's easy to remember. So now we need to look at non-coding sequences. Basically, what we explained at the start is a bit oversimplified. Not all DNA codes for polypeptides. There are non-coding sequences which have other functions. So first, we have tRNA and rRNA genes. So these make the tRNAs, right, and the rRNAs. rRNAs form the ribosome itself, right? So they make the organelle, and tRNAs are the ones that carry the amino acid. Hopefully you remember. And then we have promoters. So promoters, right, are regions of DNA that are upstream of a gene, so before the gene, and they initiate transcription, right? How does that work? Well, because RNA polymerase can bind to the promoter with the help of transcription factors, right? And then there's also, it gets a little more complicated, there's repressor sequences which block transcription by binding the transcription factor, and that then prevents RNA polymerase binding. There's also activator sequences that enhance transcription by binding the transcription factor, and then that promotes RNA polymerase binding, right? So gene expression really depends on which transcription factors are present to bind to the promoters, right? Because that allows either the RNA polymerase to bind or not, which causes transcription or not. Okay. Then we also have telomeres. So telomeres protect chromosomes. They're basically the caps of the chromosome, right? The ends. And they're only present in eukaryotes. And then introns, right, are parts of the gene that are removed from mRNA during editing because they're non-coding. So not the whole of the gene actually codes for a polypeptide. Okay, that's also quite important to remember. And one last thing about promoters is that they respond to transcription factors when bound to another molecule in some cases. So for example, that's how the testosterone receptor activates male-specific genes, okay? So normally the transcription factor needs to be bound to another molecule, and that way that allows the body to signal, right, when to activate it or not. And that allows us to change our cells on demand, or more specifically the proteome of our cells, right, which proteins we have at that given time. So it doesn't end there, because once we've actually transcribed a gene, oftentimes there's a lot of modifications that happen to the RNA before translation. This only happens in eukaryotes, okay? Important. This does not happen in prokaryotes. And this happens inside the nucleus still, right? So before the mRNA enters the cytoplasm. So what are the key modifications? Well, first of all, a 5' cap is added, capping, right? It's a modified guanine nucleotide, okay? And this stabilizes the RNA and aids further processing. We also add a poly-A tail, which basically means you add 100 to 200 adenines, just like A, right, to the 3' end, and that protects the mRNA from degradation. So both of these are to protect mRNA, basically. That's the take-home message, right? And then we also remove the introns. Remember we said introns are found in a gene and they're non-coding. So we remove these, okay? That's quite important. Otherwise, we won't form a mature mRNA. But there's more, because the introns allow for a very interesting thing, which is alternative splicing. So this allows multiple polypeptides to be made from one single gene, right? So what you can do is you can do exon skipping, which involves, for example, you take exon 1 and exon 3, but not exon 2. And then you have polypeptide B. But then you can also take the 3 of them, right? Or you take just exon 1 and exon 2, or exon 2 and exon 3. So this enables a lot of protein diversity without actually having to duplicate genes, right? Without it needing more genes, you already have a lot of protein diversity, which is super important because we need to code for lots of different functions with our DNA. Great. So now that we've looked at post-transcriptional modifications, we need to look at translation in more detail, right? Specifically how it starts. So basically what happens is that methionine is always the first amino acid. How come? Because it's attached to an initiator tRNA, which as the name says is the first one, right? So the initiator tRNA is going to bind to the small ribosomal subunit, right? And then it's going to scan for the start codon, okay? So remember, the ribosome is going to have the mRNA on it, right? And then it can move along the mRNA. So it's going to find the start codon. The start codon, by the way, is complementary to the anticodon, which has methionine. This is why this is the initiator tRNA, right? It's because it's the start codon. Okay, so once that's paired, right, then the large subunit of the ribosome joins, okay? Then this one moves on from the A site to the P site. So this is what we were talking about, but in a little more detail. So then a second tRNA can bind to the A site over here. And that again matches the next codon, obviously, right, with the tRNA anticodon. And a peptide bond is going to form between these two amino acids, as you can see here, right, between the methionine and the phenylalanine. Great. Once that happens, the ribosome shifts again, moving the initiator tRNA to the E site. And from the E site, it exits, right? Meaning now what was in the A is now in the P, and then another one can come into the A, same thing, form a peptide bond, and so on and so on. Okay. But we're not done there, because after translation, a lot of polypeptides are actually modified before they become functional proteins. So some main types of modifications, and I want you to write these down, are first, methionine removal. So the initiator one oftentimes is removed. Then also you can change side chains, so side chains of the amino acids, right? You can phosphorylate them, you can glycosylate them. Then also a lot of times you form disulfide bonds afterwards, right, to stabilize the structure. Sometimes you cleave propeptides, okay, which are parts of the polypeptide. And then sometimes also you need to fold it into quaternary structures or conjugated proteins, which if you remember from the protein topic, right, quaternary structures where you have different polypeptides joining together, and conjugated proteins is where you then have parts that are not polypeptides, such as a heme group joining as well. So here's an example, the insulin formation, right? So it's the INS gene, right, which is transcribed in the pancreas, and then the mRNA is going to be translated on the rough ER. So then we have pre-proinsulin, which is the first polypeptide. We then cleave the signal peptide from this. So then we got proinsulin, right? So this is cleaving the propeptide, remember? Then we're going to fold, right? Get 3 disulfide bonds. And also we're going to have proteases cut some parts, right? They're going to cut proinsulin into an A chain and a B chain, okay? And then there's final trimming. So there's 3 steps there, okay? First, the folding with the disulfide bonds, then the protease is cutting, and then some final trimming, and that forms what's the actual insulin that we use, right? Mature insulin, and this is held by those disulfide bonds. And last but not least, okay, then we also have amino acid recycling. So basically, some of the proteins that are formed are going to be non-functional, or they're going to be damaged, right? Or some proteins are going to be fine, but then they're no longer needed at one point, right? So then those are going to be broken down by proteasomes. So A healthy cell, right, requires constant protein breakdown and synthesis. So how does this work? Well, what happens is proteins are tagged with ubiquitin, okay? So ubiquitin is a tag that marks them for destruction, and that leads them to the proteasome. The proteasome has regulatory subunits at both ends, so here, and these recognize the ubiquitin, right? So they're ubiquitin-tagged proteins. And then we have the core parts, right? There's an ATP-dependent subunit that unfolds the proteins first. And then there's the core chamber. And this contains proteases that digest the proteins into amino acid chains, into smaller ones, right? So after final digestion, we can release the free amino acids, which can then be reused to make new proteins. So remember, to remove faulty proteins, but also to recycle amino acids from proteins that are not needed anymore. Let's do some questions. So the first one is, what does the anticodon of a tRNA molecule pair with during translation? Pause now, and I'll let you think, but I'm going to count down from 3 and say the answer. So 3, 2, and 1. Okay. So remember, the anticodon of a tRNA, and the tRNA contains the amino acid, and then that binds to a complementary codon on the mRNA, right? So this is how the genetic code is actually coded for to make polypeptide in the end. Yeah? Then, which of the following modifications occurs during post-transcriptional processing in eukaryotes? Once again, 3, 2, and 1. Okay, so we have the 5' cap and the poly A tail for stability of the mRNA. Remember, right? Removal of ribosomes, that doesn't happen because that doesn't happen during transcription, right? There's no ribosome. Binding of amino acids to tRNA, the same, that's translation. And then replication of the gene, that's, again, DNA replication, nothing to do with this. And then the final question, what is the function of a proteasome in the cell? Okay, 3, 2, and 1. It's D. Why? Okay. Synthesizing ribosomal RNA is actually transcription, so nothing to do with this. Folding polypeptides is done by these proteins called chaperones. You don't need to know that, but nothing to do with proteasomes. And then removing introns from pre-mRNA, same. That's post-transcriptional modifications. The proteasome is at the end of the pathway. It's once we've already made the proteins, it's involved in degrading them when no longer needed or problematic, right? When they're faulty, so that we can get the amino acids back and recycle them, okay? Great. So I'm aware this is a hard topic, so any questions, please do leave them in the comments. But otherwise, I'll see you next week for the next topic. Thank you, everyone.